Welcome, deep divers. Get ready for an expedition into the microscopic. Today, we're embarking on the deep dive into Lab 5, Microscope and Cell Structure.
That's right.
Consider this your definitive preparation guide, designed not just to help you navigate the lab itself, but to absolutely conquer that pre-lab quiz.
Yeah, the quiz is key.
We're on a mission to make you truly well-informed about the invisible architecture of life.
And what's truly transformative about this deep dive is how it demystifies the very tool that unlocked biology. The microscope isn't just a piece of equipment. It's a gateway.
A gateway, I like that.
It transforms invisible worlds into observable realities, allowing us to understand life's most fundamental building blocks. This conversation will equip you with the core concepts, practical steps, and critical insights needed for success.
Especially for that diagnostic pre-lab exam, right?
Exactly.
Especially for that.
So let's kick things off with a fundamental aha moment that underpins everything we're about to explore. This is where it gets truly fascinating. We're talking about cell theory.
Foundational stuff. What's revolutionary about it isn't just that all life is made of cells, but that this simple idea unified biology. Before cell theory, observations were, fragments.
All over the place, yeah.
Afterward, it transformed a collection of disparate findings into a coherent understanding, revealing a universal blueprint for life. The cell isn't merely a building block, it's the fundamental operating system.
The smallest unit that shows all the characteristics of life.
Exactly. The smallest structure that genuinely exhibits all the characteristics of life. That universality, that's the profound insight.
Building on that and connecting this to the bigger picture, this universal blueprint isn't monolithic. Not all cells are created equal, so to speak. We classify them broadly into two distinct types, prokaryotes and eukaryotes. Prokaryotic cells, like bacteria, are generally far simpler.
Simpler how?
Well, they lack a true nucleus, meaning their genetic material isn't neatly enclosed in a membrane-bound compartment. And they also lack other complex membrane-bound organelles that characterize eukaryotes.
So if I'm picturing a prokaryote, it's almost like a tiny, efficient studio apartment with everything out in the open, ready for action?
Exactly. That's a great analogy. Whereas eukaryotes are much more akin to multi-room houses, with specialized areas for specific tasks. These are the cells you find in fungi, plants, animals, protozoa, algae, and yeast.
A little more complex.
Significantly more complex, yes. Defined by having a nucleus that houses their genetic material and a sophisticated suite of membrane-bound organelles. Think of mitochondria.
The powerhouses.
The powerhouses for energy production, exactly. Or the endoplasmic reticulum, vital for protein and lipid synthesis. This fundamental structural difference isn't just about complexity, it reflects billions of years of evolutionary divergence.
Wow.
Leading to the incredible diversity of life we see today. Understanding this basic split is your first key to categorizing almost every organism on Earth.
That distinction really does feel like a pretty monumental divide. What does it tell us about the earliest forms of life, or the potential for cells to become even more complex?
It tells us a great deal. Prokaryotes represent an ancient, highly successful design, capable of thriving in incredibly diverse environments. Eukaryotes, however, evolved later, introducing internal compartmentalization.
Those rooms in the house.
Precisely. That allowed for greater specialization and efficiency. This complexity became a prerequisite for the development of multicellular organisms.
Like us.
Like ourselves, or a towering redwood, yes. In multicellular eukaryotes, individual cells tend to specialize profoundly. They might sacrifice some basic functions, like independent movement, to become incredibly good at one specific task.
Like the cells in our gut.
Exactly. Consider the cells lining your gut, specialized for absorption, or glandular cells fine-tuned for secretion, or muscle cells designed for powerful contraction.
Okay.
In contrast, unicellular eukaryotic organisms like an amoeba or a yeast cell must handle all life functions within that single complex cell.
So one cell doing everything.
Right, so you have a single highly capable cell doing it all versus a highly integrated team of specialized cells working together.
So one cell handles everything, or a dedicated team collaborates. I understand. But why does this bring us back to the microscope? Are these individual cells simply too small for us to see with our own eyes?
Precisely. Most eukaryotic cells, the ones we'll be focusing on heavily in this lab, are typically between 10 and 30 micrometers in diameter.
And a micrometer is tiny, right?
It's extremely tiny. To give you some perspective, 1 millimeter is equivalent to 1,000 micrometers. So yes, they are indeed utterly invisible to the unaided eye.
Okay.
This is why the compound light microscope isn't just a convenience. It's an essential, precision instrument that makes this entire invisible world accessible to us.
It's the gateway.
It's the gateway. And the insight here is that this isn't just a magnifying glass. It's a window into processes so delicate, so interconnected, that a tiny misalignment can completely obscure the truth.
Every part matters.
Every part, every movement has to be exact because life itself operates with such intricate precision at this scale. Without it, the very foundation of cell theory, these building blocks of life, would remain theoretical, unobservable.
Which brings us back to the quiz.
Right. For your pre-lab quiz, you might certainly encounter questions asking about the basic tenets of cell theory, or the key differences between prokaryotic and eukaryotic cells. So these definitions are absolutely crucial for your success.
We've established why this invisible world matters and the foundational rules that govern it. Now let's equip ourselves with the literal key to unlocking that world. The microscope itself.
The tool itself.
We're talking about the digital compound microscope and compound because, as we'll see, it uses multiple lenses working together. This isn't just any piece of equipment, it's a precision instrument.
It really is.
And we need to treat it with respect.
You're absolutely right. The proper use and meticulous care of the microscope are paramount, not just for its longevity and ensuring it's ready for the next person, but crucially for the quality and clarity of your own observations.
So rule number 1.
First, and this is truly non-negotiable, always carry the microscope with 2 hands. One hand should firmly grasp the arm, while the other provides stable support to the base.
Okay, arm and base. Got it.
And it needs to be carried in a vertical or near vertical position. The why here is straightforward. It's a delicate instrument full of precisely aligned lenses and intricate moving parts.
Things that can get knocked out of whack easily.
Exactly. They can be easily knocked out of calibration or damaged if it's tilted, swung, or, God forbid, dropped. It's like carrying a precious, fragile sculpture, 2 hands upright and no jostling.
That's a perfect analogy to underscore its fragility. So it's a team effort between multiple lenses to make things bigger. What are the absolute must-know components of this instrument? And what should we prioritize understanding for our quiz and effective use?
That's a great way to think about it. We can conceptualize the microscope in 3 primary zones: the viewing zone, the magnification zone, and the illumination and control zone.
Okay, breaking it down. I like that.
Understanding these broad categories can help you mentally organize the many parts. Let's start from the top, in the viewing zone. First, the ocular or eyepiece. This is the uppermost lens where you place your eyes. It always provides a 10x magnification.
Always 10x.
Always 10x for the ocular, yes. Makes the image 10 times larger right off the bat.
Okay.
Second, the head. This component structurally holds the oculars in place, allowing for comfortable binocular viewing.
Makes sense.
Third, the arm. The arm is the main structural support, connecting the head to the base and supporting the stage and all the adjustment knobs. As we mentioned, it's also the primary part you grasp when carrying the microscope.
Got it. Crucial for carrying.
Fourth, the prism slider bar. This is a crucial feature on digital compound microscopes. It's a small bar that directs the light path.
Oh, interesting. What does it do?
You push it all the way in to direct the light to the oculars for direct viewing. If you pull it out, it redirects the light to a connected computer or digital capture device.
Ah, so for saving images.
Exactly. But for your lab work, generally, you'll keep it pushed in.
Okay, that's the viewing zone. What's next?
Moving down into the magnification zone. Fifth, the nose piece. This is the revolving part to which the objective lenses are attached.
The part that clicks around.
That's the one. It allows you to easily switch between different magnification objectives with a satisfying click. Sixth, the objectives. These are the set of 4 lenses attached to the revolving nose piece.
4 of them.
Usually 4, yes. They're designed to be used in a specific ascending order of magnification. Each objective has its magnifying power clearly labeled on its side.
So what are the standard ones?
You typically have the scanning objective, usually the shortest, providing 4x magnification. Then the low power objective, typically 10x. After that, the high dry objective, commonly 40x.
Okay, 4, 10, 40.
And finally, the oil immersion objective, the longest, usually 100x. Now, a critical detail for our deep dive. The oil immersion objective is not used in BI 201.
Oh, good to know.
You won't need to worry about it for this specific lab. Don't touch it.
That's a good heads up, saving us from a potential procedural error. So if the ocular is 10x, and then we have these different objective lenses, 4x, 10x, 40x, how do we figure out the total magnification? What's the direct formula for that?
Total magnification is straightforward and absolutely essential for understanding what you're seeing. It's simply the magnification of the ocular lens.
Which is always 10x.
Always 10x, yes,
Okay.
times the magnification of the objective lens you are currently using.
Ah, simple multiplication.
Exactly. So,
But
with the scanning objective, 4x, your total magnification is 10x times 4x equals 40x.
okay.
With the low-power objective, 10x, your total magnification is 10x times 40x equals 400x.
Makes sense.
And with the high dry objective, 40x, your total magnification is 10x times 40x is 400x.
That makes perfect sense. A simple multiplication, but it's so crucial for translating what we see into actual scale. Now, alongside magnification, there are two other critical concepts tied to these objectives that we need to grasp: working distance and diameter of the field of view. What are those, and how do they change as we increase magnification?
You've hit on two very important inversely related concepts.
Inversely related. How so?
Working distance is the physical space between the tip of the objective lens and the top surface of the microscope slide. Diameter of the field of view refers to the actual circular area of light you see when looking through the microscope, essentially the visible portion of your specimen.
Yeah.
What's truly fascinating here is that both the working distance and the diameter of the field of view decrease dramatically as magnification increases.
So zoom in, things get tighter.
Exactly. Think about it. When you zoom in, that is, increase magnification, the objective lens physically moves closer to the slide, reducing that working distance.
Right, less space.
Less space. Simultaneously, you're seeing a much smaller, more detailed area of the specimen, which means the overall field of view becomes smaller.
Okay. You see less of the thing, but in more detail.
Precisely.
Okay.
While we won't get bogged down in exact numbers now, just know that at our highest power, 40x, the gap between the objective and the slide becomes incredibly tiny, often less than a millimeter.
Wow, that's close.
Very close. This inverse relationship is critical for understanding why specific focusing techniques are absolutely necessary, particularly the cautions about using the coarse adjustment at high power.
That truly drives home why precision is so paramount, especially when you're at the highest magnifications. Now, let's consider the platform where we place our samples and the controls that adjust our view.
Right. That brings us to the illumination and control zone. Seventh, the stage. This is the flat platform that firmly supports your microscope slide.
Where the action happens.
Exactly. Positioning it directly over the hole that allows light to pass through. It also features stage clips to hold your slide securely in place.
Okay.
Eighth, the mechanical stage knobs. Located typically beneath the stage on one side, these are a pair of knobs. One controls the forward and backward movement of the slide,
Front and back.
and the other controls its precise left and right movement. They allow for incredibly fine positioning of your specimen within the field of view.
Essential for finding things.
Absolutely. Ninth, the rheostat. Found usually on the right side of the base, this knob regulates the voltage supplied to the light source, thereby controlling the light intensity.
The dimmer switch.
Basically, yes. And a critical detail for preserving the expensive $50 bulb, which is prone to burning out, always use a medium rheostat setting, specifically around 4 or 5.
Not cranked all the way up.
Definitely not. Too high a setting generates excessive heat and dramatically reduces the bulb's lifespan. You certainly don't want to be the one responsible for frying a bulb for the entire class.
Good tip. Keep it moderate.
10th, the condenser. This is a specialized lens system positioned directly beneath the stage. Its job is to precisely focus the light from the illuminator onto the specimen.
So it concentrates the light.
Exactly, ensuring even and optimal illumination across your field of view. The condenser should always be kept in its highest position to achieve the best illumination.
Always up. How do we make sure?
That leads to number 11, the condenser adjustment knob. As its name implies, this knob, usually found below the stage, is specifically used to ensure the condenser is maintained in its highest, most effective position.
Okay, check that's up.
12th, the iris diaphragm. This is a small lever located directly beneath the condenser. It acts like the iris of your eye, regulating the amount of light that actually passes through the specimen.
Controlling brightness again. How's it different from the rheostat?
Good question. The rheostat controls the intensity of the light source itself. The iris diaphragm controls how much of that light actually reaches the specimen. It's more about adjusting contrast.
Okay. Intensity versus amount.
Precisely. Too much light can wash out delicate details, making the specimen appear transparent, while too little makes the image dark and difficult to discern.
Got it. Find the sweet spot.
13th, the coarse adjustment knob. This is the larger focusing knob. It's designed for preliminary focusing by making relatively large and rapid movements of the stage, either raising or lowering it.
The big knob for big moves.
Right. But crucially, you should only use the course adjustment knob with the 4x scanning and 10x low power objectives. This is a fundamental safety rule.
Only low power. Why?
We'll get to that, but it relates directly back to that small working distance at high power.
Okay, hold that thought. What's the other knob?
14th, the fine adjustment knob. This is the smaller, more sensitive focusing knob. It's used for final or fine focusing by making very subtle adjustments to the stage's height.
The small knob for small tweaks.
Exactly. This knob becomes your best friend and your only focusing tool at higher magnifications because of that minimal working distance we keep mentioning.
Wow, that's a comprehensive tour of the microscope's anatomy. It's clear how knowing these parts by name and function is going to be incredibly important for that pre-lab quiz.
Absolutely.
You'll definitely want to know how total magnification is calculated for different objectives and deeply understand that inverse relationship between magnification, working distance, and field of view.
Think of this foundational knowledge as your pre-flight checklist before taking off into the micro world. It's essential.
We've just learned the intricate mechanics of our microscope. Now, for the core of our discussion, translating that knowledge into hands-on mastery.
Putting it into practice.
This section will walk us through the practical steps of using the microscope effectively, using the classic letter E slide as our initial guide. This is where we focus heavily on procedure and all those critical safety cautions, so pay extra close attention.
This is where theory meets practical application, and every step matters. Let's start with the setup and initial focus using that familiar letter E slide.
Okay, step 1.
First, and this is an absolute must, always clean the microscope lenses with lens paper. Very specific.
Lens paper only.
Only lens paper. Do not use chem wipes, regular tissues, paper towels, or any cloth, as these materials can scratch the delicate lens coatings.
Got it. Protect the lenses.
Next, ensure your microscope is powered up. Plug the round power cord into the back of the microscope and into an outlet. Then connect the square end of the USB cable into the microscope's back and its small flat end into the UFD port on your computer, like an iMac, if you're using the digital functionality.
Power and USB. Check.
Before you even touch the power switch, make sure the rheostat is turned all the way down to its lowest setting, 1.
Ah, that bulb-saving tip again.
That bulb-saving tip. This simple step is vital to protect the bulb from a sudden power surge when you switch it on.
Okay, rheostat to 1.
Also, confirm that the prism sliding rod is pushed all the way in if you intend to view directly through the oculars, rather than routing the image to the computer.
Slider in for viewing.
Right. Now, turn on the power switch for the light and gently adjust its intensity to a medium setting, specifically around 4 or 5 on the rheostat.
Medium light, 4 or 5.
A critical caution I cannot overstate. Higher settings generate excessive heat, drastically shortening the lifespan of that costly bulb. Moderation is key here.
Be kind to the bulb.
Then, rotate the nosepiece until the 4x scanning objective clicks firmly and precisely into place. Start low.
Always start with the lowest power, 4x.
Always. You'll need to adjust the interpupillary distance, the space between your eyes, to comfortably view through both oculars. Look through both, and gently pull them apart or push them together until you perceive a single, perfectly round field of view.
Get rid of the double vision.
Exactly. It's helpful to know your comfortable interpupillary distance on the scale provided for future labs.
Good idea.
Now, place your E slide on the stage. For your initial viewing, orient it so you can read the letter E properly with your unaided eye. Use the mechanical stage knobs to carefully center the E directly over the hole in the stage where the light passes through.
Center the E.
Here's a crucial safety step. While watching from the side, with your eyes at the level of the stage, gently raise the stage until it stops moving, by slowly turning the course adjustment knob away from you.
Okay, wait. Watch from the side while raising the stage. Why?
This is a non-negotiable caution. Always visually monitor the stage from the side when raising it to ensure the objective lens doesn't crash into the slide. It prevents damage to both. Very important.
Ah, okay. Avoid collision. Makes sense.
Once the stage is fully raised, shift your gaze to look through the ocular. Now, slowly lower the stage by turning the course adjustment knob toward you. Continue lowering until the E comes into a rough focus.
Lower slowly while looking through the eyepiece.
Right. Then, for sharp, crystal clear focus, use the fine adjustment knob. Another important caution. Never turn the fine adjustment knob more than about half a turn in either direction once you've achieved that rough focus. It's designed for subtle, minute changes.
Fine-tune with the fine focus. Small movements.
Finally, while still looking through the oculars, adjust the iris diaphragm lever to optimize the image contrast. This will enhance the visibility of details without washing them out.
Adjust contrast with the iris. Got it. That's a lot of meticulous steps to get started. Once we have our initial focus at this low power, 4x, what are the key observations we should be making?
The very first, and often most surprising observation, is image inversion.
Inversion, like upside down.
Upside down and backward. You'll notice that the letter E appears completely reversed compared to how it looks on the slide with your unaided eye.
Oh, okay.
This is a fundamental characteristic of compound microscopes that you must always keep in mind. It means that if you move the slide to the left on the stage, the image you see will move to the right.
Opposite world.
Exactly. If you move the slide away from you, the image will appear to move toward you. This can be disorienting at first, and it genuinely takes some mental rewiring.
I bet.
But with practice, it becomes intuitive. As part of the lab, you should draw the E, as seen by the unaided eye, then as it appears at 4x total magnification, and again at 10x total magnification, to visually track this inversion and the increasing magnification.
So if I'm trying to chase down a tiny moving organism, I have to mentally reverse my movements. That definitely sounds like a skill that takes practice. Now, how do we move to higher magnifications effectively without losing our specimen?
The process for transitioning to higher magnifications is quite systematic and relies on two critical properties of the microscope. First, before rotating the nosepiece, you must ensure the letter E is precisely centered in your field of view under the scanning 4x objective.
Center it first. Why is that so crucial?
This is absolutely critical because the microscope objectives are paracentric. In simple terms, paracentric means that if your object isn't perfectly centered at a lower power, it will move out of the field of view entirely when you switch to a higher magnification.
Ah, because the field of view shrinks so much.
Precisely. It makes it incredibly difficult to relocate. So the big question becomes, what happens if your object isn't centered? It simply won't be there when you zoom in, and you'll be scanning an empty field. Frustrating.
Ah, so centering is the golden rule before increasing power, like lining up for a perfect shot before zooming in with the camera, otherwise you'll just have a blurry mess of the background.
Exactly. Once centered, rotate the nosepiece to bring the 10x low power objective into position. You'll hear and feel it click firmly into place.
Click, okay.
Look through the oculars and slowly turn first the coarse and then the fine adjustment knob away from you until the image becomes sharp.
Coarse, then fine at 10x.
Yes, at 10x, you can still use the coarse knob carefully for initial focus, then fine-tune. But a critical caution here. After this initial rough focus at 4x and 10x magnification, you should only use the fine adjustment knob to sharpen the focus.
Okay, here comes. Why only fine focus after 10x?
Because using the coarse adjustment at 40x or higher can very easily result in the objective lens crashing into and damaging the slide, the lens, or both, due to that greatly reduced working distance we talked about.
Got it. Coarse adjustment is forbidden at 40x. Fine focus only.
Absolutely forbidden. Now, to move to the 40x high dry objective, you'll slowly rotate the nosepiece. As you do this, once again, watch from the side with your eyes at the level of the stage.
Watching from the side again.
Yes, to be absolutely certain that the high-power objective does not contact and break the slide. This is another crucial safety measure that prevents costly damage.
Check clearance before clicking into 40x.
Right. Once the 40x objective is in place, you'll notice that the objectives are parfocal.
Parfocal. What does that mean?
In simple terms, parfocal means they are pre-aligned so that only very minor fine focusing is needed when you rotate from one objective to another.
Oh, nice. So it should be almost in focus already.
It should be very close.
Yeah.
You shouldn't need to make drastic adjustments with the fine focus knob to bring the image back into sharp focus.
That parfocal feature sounds like a real time saver, a thoughtful design choice. So once we're at 40x, is it exclusively the fine adjustment knob, no matter what?
Precisely. Once you're observing an object under high power, 40x, only the fine adjustment knob should ever be used for focusing.
Got it. Locked in.
It's also important to be aware that very thick preparations simply cannot be observed under high power due to that extremely small working distance we discussed earlier.
Right, the lens can't get close enough to focus through thick stuff.
Exactly. The lens physically won't be able to get close enough to the specimen to focus through its thickness effectively.
That makes a lot of sense. The thinner the sample, the clearer the view at high power. This brings us neatly to another phenomenon directly impacted by magnification, something called depth of focus. Expert T, how does this play out when we're peering into our specimens, and how do we even begin to observe it?
That's an excellent question, as depth of focus is often misunderstood. Depth of focus refers to your ability to see through the thickness of a specimen, to clearly resolve objects that are at different vertical levels within the sample.
Seeing in 3D, sort of.
Kind of, but it becomes much more apparent, or rather more limited, at higher magnifications. To observe this, we use the thread slide, which contains colored threads layered over one another.
Okay, the thread slide.
Place this slide on the stage and start with a scanning objective, 4x, and focus. You'll likely see all three threads somewhat in focus, then move to 10x, and finally to 40x.
Following the same procedure, centering and focusing?
Exactly. At high power, 40x, you'll focus on the exact point where the threads cross. Then, using only the fine adjustment knob, you'll slowly adjust it slightly up and slightly down.
Rocking the fine focus back and forth.
Gently, yes.
Yeah.
You'll notice that only one of the threads will be in crisp, sharp focus at any given moment. The others above or below it will be blurry.
Ah, so you can tell which one is on top.
Precisely. By making these minute adjustments, you can mentally slice through the specimen's thickness, bringing each individual thread into clear focus one at a time. This allows you to determine which color thread is at the bottom, which is in the middle, and which is on the top.
That's cool.
This exercise beautifully illustrates how depth of focus decreases dramatically as magnification increases. The narrower the depth of focus, the finer your ability to distinguish different planes within a specimen.
That's a fantastic interactive way to visualize how magnification impacts what you can see. For the pre-lab quiz, I would definitely expect questions on the sequential steps of focusing, all those critical cautions like watching from the side when raising the stage or rotating objectives, and crucially, when to use coarse versus fine adjustment.
Absolutely key points.
And absolutely, be ready to explain image inversion, parfocality, paracentricity, and how depth of focus changes with magnification. These are all prime quiz material, and understanding why these phenomena occur is just as important as knowing what they are.
Indeed, mastering these operational nuances is the hallmark of a confident microscopist. And likely quiz questions.
We've mastered the mechanics of the microscope. Now, before we can explore the fascinating world of plant and animal cells, we need to get them ready for viewing. Let's talk about preparing a wet mount.
Ah, yes, the wet mount. A fundamental and thankfully simple technique for viewing fresh, often living material. It's a temporary preparation used to observe specimens suspended in a liquid.
Sounds straightforward. How's it done?
The procedure is quite straightforward. First, take a clean microscope slide and place a single drop of water, or another liquid medium, directly in its center.
1 drop of water.
Next, carefully place your specimen, which should be very thin, into that drop of water.
Specimen in the water.
Then, take a cover slip. Hold it at a 45-degree angle, touch one edge of the cover slip to the edge of the water drop,
Angle it down.
and then gently lower it slowly over the specimen. The goal here is to avoid trapping air bubbles, which will appear as distracting circles of various sizes with very dark edges under the microscope.
Lower it slowly, like closing a door, not dropping it flat.
Exactly. Slow and steady wins the race here to achieve a bubble-free mount.
That technique makes good sense. But a common query we hear is, why is a cover slip actually necessary? Is it just to keep the water from drying out?
That's certainly one benefit. It slows evaporation, but it serves several crucial purposes that are vital for both optimal viewing and protecting your equipment.
Okay, what else does it do?
The cover slip. First, protects the objective lens. It creates a vital physical barrier between the specimen and the delicate, often expensive objective lens.
Prevents scratches.
Prevents direct contact and potential scratching or contamination, especially those high-power objectives that get so close. Second, it flattens the specimen.
Makes it thinner.
It gently compresses the specimen, creating a more uniform thickness. This is crucial because light needs to pass through evenly, and a flattened specimen ensures most of your sample is within a similar focal plane, making it easier to focus.
Oh, easier focusing.
Third, it holds the water and specimen in place, prevents things from drifting around. And fourth, related to flattening, it creates a uniform focal plane, minimizes height variations, allowing for a clearer view, especially at high magnifications where depth of focus is very limited.
So it's far more than just a simple lid. It's an essential component for both optimal viewing and microscope protection. Many cells we'll be looking at are quite transparent, right? How do we make those delicate internal structures more visible?
You're absolutely right. Many unstained cells can be exceedingly difficult to see due to their transparency. This is where staining comes in.
Adding color.
Essentially, yes, stains are biological dyes that specifically bind to and add contrast to certain cellular components, making them stand out against the background.
Okay, how do we stain a wet mount without messing it up?
The general technique, without disturbing your perfectly placed cover slip, involves placing a small amount of stain next to one edge of the cover slip.
Drop of stain beside the cover slip.
Then you place a piece of blotting paper or a paper towel on the opposite edge of the cover slip.
Paper towel on the other side.
This action draws the water from under the cover slip through capillary action, which in turn pulls the stain underneath, allowing it to interact with and colorize your specimen.
Yeah, that's clever. It wicks the stain under.
It's a neat trick that ensures minimal disruption to your prepared slide.
That is very clever. For the pre-lab quiz, I anticipate questions about the sequential steps to prepare a wet mount and, perhaps more importantly, why a cover slip is used, outlining its multiple functions.
Definitely need to know the why.
Also, be prepared to explain the general purpose of staining specimens, not just what it does, but why we do it. Visibility and contrast.
Those are indeed excellent points to focus on for your preparation. Understanding the rationale behind each step will solidify your knowledge.
Now, let's bring it all together and apply our newly honed microscope skills and preparation techniques to distinguish between different types of cells.
The fun part.
Get ready for some aha moments as we compare and contrast plant and animal cells under the lens.
This is where we start seeing the incredible diversity and specialization of life at the cellular level. Let's dive into plant cells first.
Okay, plants first. What are we looking at?
For Aleolodia leaf cells, you'll prepare a simple wet mount using a small leaf torn from a Melodia sprig that's a common aquatic plant.
Like from a fish tank.
Often, yes. When you examine it under the microscope starting at 4x and moving up to 40x, you'll observe small, distinct, and typically box-like units. Each is an individual plant cell.
Little green boxes.
Pretty much. What's particularly fascinating is that as the leaf warms slightly under the microscope light, you may actually see the small, green, oval-shaped chloroplasts.
The photosynthesis factories.
Exactly.
Yeah.
You might see them moving in a directed circular flow within the cell. This movement is due to cytoplasmic streaming.
Cytoplasmic streaming, what's that?
It's an active circular flow of the cytoplasm that speeds up the distribution of nutrients, organelles, and other materials throughout the large volume of the cell, like an internal transport system.
Cool, like a little cellular current.
Precisely. When drawing an Melodia cell, you should focus on accurately labeling the distinct cell wall, the outer box, the inner cell membrane, the prominent large central vacuole, which takes up a lot of space, the chloroplasts, the general cytoplasm, and indicating the direction of cytoplasmic streaming with arrows.
Lots to label there. Cell wall, membrane, vacuole, chloroplasts, cytoplasm, streaming.
Next, we move to onion epidermis cells. You'll again make a wet mount, but this time using a thin, transparent piece of epidermis carefully peeled from the inner surface of an onion leaf.
The thin skin inside the onion layers.
That's the one. Because onion cells, unlike Alodia, do not contain chloroplasts and are quite transparent, staining with iodine is absolutely crucial here.
Why iodine for onions?
We use iodine because it enhances the visibility of cell structures, particularly the nucleus, by reacting with starch, sometimes present. You'll add the iodine using that blotting paper technique.
Wick it under again.
Right. Under magnification, you'll observe elongated, somewhat rectangular cells forming a tight brick-like pattern.
Like bricks in a wall.
A good comparison. You'll be able to clearly distinguish the large central vacuole from the surrounding cytoplasm, and with good focus, you might even spot the nucleolus inside the nucleus.
Nucleolus within the nucleus.
If you're lucky and have good focus. For your drawing, focus on labeling the strong cell wall, the cell membrane, the large central vacuole, the cytoplasm, the prominent nucleus, and the nucleolus if you observe it.
Similar labels, but no chloroplasts this time.
Finally, for plant cells, we'll investigate potato cells. You'll prepare a wet mount of a very thin section of potato, carefully sliced using a razor blade.
Thin slice of potato.
Very thin. Place this potato section into the water drop and add a cover slip. You'll then stain the potato cells with iodine, again by adding it next to the cover slip and drawing it under.
Iodine again. What does it show in potatoes?
When examining the potato at 4x, then 10x, and finally 40x power, you'll observe the cells densely filled with dark staining often oval-shaped organelles called amyloplasts.
Amyloplasts. What are they?
They are specialized organelles for starch storage. And this brings us to a crucial point. What color are the amyloplasts after iodine staining, and what does this vivid color indicate they contain?
Okay, thinking. Iodine stands starch, so they should turn dark.
Exactly. The very dark, almost black color indicates the abundant presence of starch, as iodine reacts strongly and specifically with starch molecules, turning them a deep blue-black.
Ah, so potato stores lots of starch in these amyloplasts.
Tons of it. Due to the sheer number and size of these amyloplasts, it can be quite difficult to clearly observe the nucleus and vacuole in potato cells.
They're kind of hidden by the starch bags.
Pretty much. Your drawing should label the cell wall, cell membrane, cytoplasm, and the very prominent amyloplasts.
So we're seeing some common plant cell features like rigid cell walls and large central vacuoles, but also specialized structures like chloroplasts for photosynthesis in Elodia and amyloplasts for starch storage in potatoes. That's a great demonstration of how plant cells differentiate. Now let's jump to animal cells.
For animal cells, we'll examine human cheek cells. This involves a simple wet mound created from a gentle scraping of the inside of your cheek using a clean toothpick.
My own cheek cells. Cool.
Yep. Stir the collected scrapings into a drop of water on the slide and carefully add a cover slip. Under the microscope, these cells are typically thin, flat, and somewhat irregular in shape.
Not boxy like plant cells.
Not at all, and they tend to clump together. They're also usually quite transparent, so staining with methylene blue is absolutely essential for visibility.
Methylene blue this time. What does it stain?
Methylene blue's purpose is to stain cell nuclei and other acidic components, making them visible, often a distinct blue color.
Okay, highlights the nucleus.
What's truly fascinating here is how these cells, despite being so simple in appearance, directly reflect their specialized function for protection. They form a protective layered barrier in your mouth.
Flat and layered for protection. Makes sense.
You might also notice very small dark blue rods or spheres covering some cells. These are bacteria.
Bacteria on my cheek cells.
Totally normal. They are naturally present and usually harmless inhabitants of the human mouth. When drawing a cheek cell, label the outer cell membrane, no cell wall, the internal cytoplasm, the central nucleus, and any bacteria you observe.
It's incredible how, even at this basic microscopic level, we can clearly see the functional differences between cells. So for our drawings and labeling, what's the most important takeaway for distinguishing plant and animal cells? What are the key visual cues?
The comparative analysis is absolutely crucial for your understanding and for the quiz. You must be able to visually distinguish between plant and animal cells based on their observable structures.
Okay, the big differences.
For plant cells, the required labels include the cell wall, cell membrane, large central vacuole, chloroplasts, cytoplasm, nucleus, amyloplasts, and indications of cytoplasmic streaming. For animal cells, you'll focus on labeling the cell membrane, cytoplasm, nucleus, and any bacteria you observe.
So the key plant-only structures are?
The key distinguishing features are that the rigid cell wall, the energy-producing chloroplasts, the starch-storing amyloplasts, and the expansive large central vacuole are unique to plant cells and conspicuously absent in animal cells.
And animal cells.
Animal cells, on the other hand, often exhibit more irregular shapes and lack these rigid structural components and specialized organelles like chloroplasts or amyloplasts. They just have the cell membrane on the outside.
Got it. Cell wall, chloroplasts, large vacuole, amyloplastics was plant. Irregular shape, membrane-only animal.
That's a good summary. For your pre-lab quiz, you absolutely must be able to distinguish between plant and animal cells based on these observable structures. Expect questions asking what specific structures iodine or methylene blue highlight, and to identify the primary function of organelles like chloroplasts and amyloplasts.
That comparison chart sounds essential.
Creating a detailed chart comparing these cell organelles will be an invaluable study tool here. Absolutely.
We've covered a vast amount of ground, from the smallest cell to the intricacies of the microscope. Now, let's bring it all home and specifically tailor this conversation for your pre-lab quiz success.
All right, let's focus on acing that quiz. Remember, knowledge is most valuable when understood and applied, and that profoundly includes respecting your tools and preparing diligently.
Starting with putting the microscope away properly.
Absolutely. First, a quick but crucial review of proper microscope storage. This isn't just a formality. It directly reduces the chances of damage and ensures it's ready for the next user.
Okay, the shutdown procedure.
When putting it away, you should ensure the prism sliding bar is pushed all the way in.
Slider in.
Confirm there's no slide left on the stage. Easy mistake to make.
Check the stage. Clear.
Lower the stage, fully using the course adjustment knob, prevents stress, protects lenses,
Stitch all the way down.
retract the mechanical stage so it's not sticking out, less likely to get bumped.
Knob centered.
Rotate the nose piece so the 4x objective is in the viewing position. Shortest objective, safest position.
Back to 4x.
Return the rheostat to its lowest setting. 1. Protect that bulb.
Rheostat to 1.
Finally, ensure the power switch is off.
Power off. Done. Those storage steps really underscore the idea that proper handling is an integral part of being a responsible scientist, respecting your tools and your peers. Now let's talk about anticipating quiz questions. The lab materials often include questions for further thought and review that practically scream quiz material. Let's tackle them one by one, explaining not just the answers, but the underlying concepts you need to grasp.
Excellent idea. These questions are prime candidates for your pre-lab quiz, designed to test your understanding of core concepts. Let's run through them. Question 1. Why must you center and focus the object in the field of view under low power before switching to high power?
We covered this. It's about that property.
Acortocentric. The answer here connects directly to the paracentric property. As you increase magnification, the field of view drastically shrinks.
So if it's not centered, poof, it's gone.
Exactly. If your object isn't centered under low power, it will simply move out of the much, much smaller field of view when you switch to high power, making it impossible to find. Centering ensures it stays visible. Question 2. Why is only the fine adjustment used for high power?
The danger zone. It's about the working distance, right?
Spot on. This relates to 2 critical factors: the extremely small working distance at high magnification, 40x, and the increased sensitivity of the focusing mechanism.
The lens is super close to the slide.
Incredibly close, maybe 0.5 millimeter. Using the course adjustment risks smashing the objective into the slide. Catastrophic.
So fine focus only for safety and precision.
Precisely. The fine adjustment allows for subtle, precise focusing within that very narrow focal plane without risking any physical contact. Question 3. Describe how each of the following is affected by switching from the low to high power objective. Total magnification, brightness of field, diameter of field, working distance, depth of focus.
Okay, this is the big relationship question. Let's break it down. Total magnification.
Increases significantly, goes up. 100x to 400x.
Brightness of field.
Decreases. Image appears dimmer as light spreads more.
Diameter of field.
Decreases dramatically. You see a smaller area.
Working distance.
Decreases significantly. Lens gets closer.
Depth of focus.
Decreases dramatically. See a thinner slice.
So magnification goes up, everything else goes down, mostly?
That's the general trend, yes. Understanding these inverse relationships is crucial. Question 4. Why must a specimen viewed with a compound microscope be very thin? Why are specimens sometimes stained with dyes?
2 parts. Thinness first. Why thin?
Simple physics. Light needs to pass through it for you to see it with a light microscope. Too thick, light gets blocked.
No light, no image. Makes sense. And staining.
Staining is for contrast. Many cells and their internal structures are naturally transparent or nearly transparent.
Hard to see clear things.
Exactly. Stains increase contrast by selectively binding to different components, making them visible and distinguishable. Without staining, you might just see a faint outline. Question 5. Human cheek cells are epithelial cells, which are highly specialized for protection. List some of the functions that these cells cannot carry out.
Okay, cheek cells protect. What can't they do? Links back to specialization.
Right. Since they're specialized for protection, they've given up or don't perform many other functions.
Yeah,
Example.
They can't move around on their own.
Correct. No motility. What else?
They don't digest food like an amoeba.
No large-scale digestion. They don't do photosynthesis, obviously.
Like no chloroplasts.
They're not specialized for absorption like gut cells or secretion like Klang cells. They don't conduct nerve impulses or perform muscle contraction.
So basically, anything highly specialized that isn't protection.
That's a good way to put it. Question 6. Complete the following chart. Cell organelle or structure, found in plant, animal, or both, function.
The big comparison chart. This is key study material. Let's hit the main ones. Nucleus.
Both plant and animal. Function. Control center holds DNA, regulates cell activities.
Cell wall.
Plant only. Function. Rigid outer layer for support, protection, shape.
Chloroplast.
Plant only. Function. Site of photosynthesis. Converts light to energy.
Amyloplast.
Plant only. Function. Starch storage.
Cell membrane.
Both. Function. Regulates what enters leaves the cell. The gatekeeper.
Cytoplasm.
Both. Function. Jelly-like substance filling the cell, suspends organelles, site of metabolic reactions.
Mastering that chart seems essential for identifying cells.
Absolutely critical.
Those answers are incredibly thorough and really highlight the key concepts that someone needs to know. But knowing the answers is one thing, acing that pre-lab quiz is another. Let's dedicate a bit more time to strategy. We've just reviewed questions that are practically guaranteed to show up. But how do you prepare mentally, and what's this cheat sheet all about? This is where we distill everything into an action plan for your success.
Exactly. The pre-lab quiz is designed to be short and focused. Typically, it includes 2 short questions, each worth 1 point, and you'll generally have about 5 minutes to complete it.
5 minutes, 2 questions. Okay.
Its purpose is multifaceted. It genuinely encourages you to focus on what you're supposed to be learning before the lab. It stimulates scientific thinking by asking you to apply concepts.
Makes you read the lab beforehand.
Exactly. And crucially, it helps you identify any confusion before you even start the hands-on lab. This way, you can seek help and clarify things up front. It's a low-stakes check-in.
And there's a specific tool for this quiz designed to actually help you perform better, right? This cheat sheet index card.
Yes, you'll be provided with a standard 3x5 or 4x6 inch index card for the quiz. This card is indeed your personal cheat sheet, but it's more accurately a study aid.
Okay, a study aid. How does it work?
The rule is you should fill it out by hand with notes, tips, formulas, and any useful information you've gleaned from reading the lab material ahead of time, which, as a reminder, you should always do.
By hand is key.
Yes, the act of handwriting those notes itself is a powerful study technique, forcing you to summarize and synthesize. You'll then return this card to your instructor together with your completed quiz.
So it helps you study and helps during the quiz.
It's a fantastic way to consolidate your learning, identify the most important information, and have key facts readily accessible under a bit of pressure.
So how do you make the most of that index card? What should go on it?
Think of it as your personal filter. What are the 5-7 absolutely non-negotiable facts or critical procedures you need to recall under pressure?
Like the magnification formula.
Definitely. Or the sequential steps of focusing. The crucial safety cautions, like when to use coarse versus fine adjustment.
Coarse only at 4x, 10x, fine only after that.
Perfect example. Or perhaps the key distinguishing features between plant and animal cells. Don't just copy verbatim from your notes. Synthesize. Write down the why behind the procedures, or a mnemonic for remembering the inverse relationships.
Like high power ACD, AFOV, 8 bright, A doof.
Exactly like that. Or a reminder, watch from side. These pre-lab quizzes count for a significant 10% of your final lab exam grade.
10% is a lot.
It is, so preparing for them diligently and using that index card effectively is a strategic investment in your overall success in the course. Use this entire section of our deep dive as your ultimate study guide. The review questions and the organelle chart are prime candidates for distillation onto your index card. Understand the why behind each concept, not just the what.
So what does this all mean for you, our listener? You are now equipped with the comprehensive knowledge to confidently approach Lab 5, deftly understand your microscope, accurately identify key cell structures, and most importantly, crush that pre-lab quiz.
You've got the tools now.
I'm genuinely excited for you to dive into the visible and invisible wonders of the microscopic world. It's a journey that truly changes how you see life itself.
And if we connect this to the bigger picture, understanding these fundamental cell structures through the compound light microscope is truly just the first magnificent step.
Just the beginning.
Just the beginning. Imagine how advancements in microscopy, such as electron microscopy, which uses electrons instead of light, to achieve vastly higher resolution.
Seeing even smaller things.
Much smaller. Or cutting-edge super-resolution techniques which push past the traditional limits of light, continue to transform our understanding of cell function and disease. We're moving beyond what we can see with a simple compound light microscope into realms that reveal molecular interactions and dynamic cellular processes in unprecedented detail.
Wow.
How might these new ways of seeing push the boundaries of medicine and biology even further, far beyond the initial observations we've discussed today? Keep that curiosity alive and keep exploring.